Hi everyone. Today I am coming with another set of questions from a second year chapter, that is biotechnology, its principles and processes. What is biotechnology? I think you are all familiar with the biotechnology. That is a common term. Biotechnology is the integration of natural science and organisms, cells, parts thereof, and molecular analogues for products and services. So that is the definition given in your textbook for biotechnology. And biotechnology has got two core techniques. The modern biotechnology has got two core techniques. What are they? One is genetic engineering and other is maintaining a sterile ambience or a sterile medium so that only the desired microbe will be able to grow and we can isolate them. So here in this chapter we mainly discuss the different tools that are required in genetic engineering and also the process of genetic engineering. What is actually genetic engineering? It mainly deals with recombinant DNA technology. So by tools of genetic engineering, we mean the tools of recombinant DNA technology. And also later on we will deal with the process of recombinant DNA technology. In recombinant DNA technology, as you know, we are trying to introduce a desired gene into a host cell. The desired gene can be called the donor DNA. It can be incorporated into a host cell. Then only we can make copies of it, or then only we can do the cloning. So for incorporating into a host cell, we need a vector. A vector, usually it will be a plasmid or a bacteriophage. So here the donor DNA is combined with the vector and that is called recombinant DNA. Okay, so this recombinant DNA, once formed, it will be introduced into the host cell, and this technology of getting a desired product from that cell, that is called genetic engineering. That is the essence of recombinant DNA technology. Okay, so this was only an intro. Now we move on to the questions. So here we have the first question for you. The role of two enzymes identified in bacteria. One of them dash and while the other will do dash. Okay, so here two sets of enzymes have been identified in bacterial cell and they are asking about the role of these two enzymes. In 1963, two enzymes were identified. Okay, that will protect that cell from the attack of bacteriophage. That will protect that bacterial cell from the attack of bacteriophage. To know more about this, we have a diagram. So in this diagram, you can see the diagram over here. In this diagram, you can see the bacterial cell which is infected by the bacteriophage. So usually bacteriophages, they are the viruses which attack the bacteria. So here you can see certain enzymes are there. These enzymes are called restriction enzymes. And these restriction enzymes, they are destroying the entered viral DNA. The bacteriophage DNA will be destroyed by certain enzymes and these are called restriction enzymes. So one of the enzyme will cut the viral DNA. You got it? One of them will cut the viral DNA and there will be another enzyme which will methylate the host DNA. Host DNA will be methylated so that this enzyme will not cut the host DNA. So methylation, once it is done, this host DNA is getting protected. So there are two enzymes. One will methylate the host DNA and the other will cut the viral DNA or DNA of the bacteriophage. So moving on to the question now. The role of the two enzymes identified. Okay. So our answer will be option D over here. So one of them will add methyl groups to the host DNA and the other will cut the bacteriophage DNA. So that is our correct answer. Okay. And by cutting the bacteriophage DNA, actually there will be degradation of the viral DNA or bacteriophage DNA. You got it? So answer for this question will be option D. Now we move on to the second question. Select the one which is not applicable for EcoRI. What is EcoR1? Okay, so what you can say about these statements? EcoR1 comes from Escherichia coli RY13. That is a strain of E. coli bacteria from which you are getting this meat. That is why it is named as EcoR1. Okay, then it will find a specific recognition sequence and it will bind to the DNA and it will form sticky ends. As all of you know, these are restriction enzymes. They will cut specific sequences in the DNA. And these specific sequences are called palindromic sequences. You know what are palindromes? In our language, in our English language, Malayalam, madam, little, like that, a lot of words are there. When you look at the letters from left to right and also from right to left, they are in the same sequence. Okay, like that, in our biology also, when you consider the DNA molecule, there are certain sequences in the DNA where you will find the same set of nucleotides from 5' to 3' as well as on the opposite strand, complementary strand also from 5' to 3'. The same sequence will be there, and these sequences are called palindromic sequences. These sequences are called palindromic sequences. So here, EcoR1 is a type of restriction enzyme which will cut at palindromic sequence. It will find specific recognition sequence and it will bind to the DNA and it will form sticky ends. It will form sticky ends. We have a picture of cutting of this EcoR1. You know, EcoR1 will identify a specific sequence from 5' to 3' GAATTC. Okay, so here EcoR1, if it finds a sequence like this, it will cut between G and A. On both the strands between G and A, it will make a cut. Okay, so here the vector of DNA will be cut with EcoR1 and also the foreign DNA is also cut with the EcoR1. So that you can see the sticky ends produced are complementary with each other and they can be joined to get a recombinant DNA like this. So remember, EcoR1 will identify specific sequences or palindromic sequences in DNA. Moving back to our question now. We have the question here. I have explained option A and B. What about C? It will cut the two strands of the double helix at specific points. I told you in between G and A it will make a cut in their sugar phosphate backbone. So that is a correct statement. And which is a wrong statement here? That is option D. In option D it is written it was the first restriction endonuclease to be identified. Okay, that is a wrong statement because hint 2, it is the first restriction endonuclease to be identified. Okay, now we move on to the next question. We have the key tools in recombinant DNA technology are, you know, recombinant DNA technology requires restriction enzymes. That is one of the main tools. They are the molecular scissors which will cut the DNA. Another one is cloning vectors. We need cloning vectors. That is another tool for recombinant DNA technology. And another tool will be a competent host. We require competent host also. So here, among options, you can find restriction enzymes and ligases. Ligases, you know, of course, to form a recombinant DNA, you have to glue the sticky ends, or even if they are blunt ends, you have to glue them by using an adapter molecule. So here, restriction enzymes and ligases, they are the main tools in recombinant DNA technology. You got it? What about other options? Other options, exonucleases are cutting the DNA at the peripheral nucleotides. Endonucleases will cut the DNA within. It will cut the DNA within. Then DNA polymerase, it is used for polymerization of the DNA or essential for DNA replication. Then reverse transcriptase. Reverse transcriptase, it will help in forming a DNA from RNA. From RNA to DNA, we require reverse transcriptase enzyme. Okay, So all these options, what about RNA polymerase? It is for transcription. That is the main enzyme required for transcription. So we have the answer now. Before that, we will have a slide here. This slide will show how exactly the restriction enzymes and ligases are working. Okay. So restriction enzymes, they are cutting a palindromic sequence and sticky ends are formed here. Okay. Now with the help of a ligase, you can attach these sticky ends. So sticky ends, they will be sealed by ligase. Ligases, it will help in sealing the sticky ends. So shall we move to our next question? Before that, our answer comes here. So our answer will be option A. So we have the next question. Sticky ends produced can join only if, as you know, in recombinant DNA technology, restriction enzymes, we are cutting the donor DNA as well as the vector DNA. We have to use the same restriction enzyme on the donor DNA as well as on the vector DNA, because then only we will get complementary type of sticky ends, and only when the complementary sticky ends are produced, they can be joined with the help of ligase. So remember, both donor DNA as well as the vector DNA, or both foreign DNA and the vector DNA, is cut with the same restriction enzyme. Then only we can produce sticky ends. Sticky ends. Sticky ends mean the ends which are complementary with each other, and they can be joined. Okay, so answer will be option A here. We have a diagram for explanation in your NCRT. You can go through this diagram. It is clearly written here that both the donor and the vector is cut with the same restriction enzyme. Okay, then we will join the donor with the vector. Then only we can produce a recombinant DNA. So sticky ends can be produced only when we cut donor as well as vector with the same restriction enzyme. So coming back to the question, our answer will be option A here. Reverse transcriptase. It is used for reverse transcription, not applicable for this recombinant DNA technology. Now, then foreign DNA and vector DNA got from the same host cell. Always it will be from different cells, foreign DNA and vector DNA we are using. Then restriction enzymes are extracted from the same microbe. Not necessary. Okay. So here our answer will be option A only. Moving back to our question. Next question. Next question is on the slide now. Yes, here it is. It is about gel electrophoresis. You know about gel electrophoresis. After cutting the DNA, we have to separate the DNA fragments. Only when you separate the DNA fragments, it can be used for genetic engineering. So the technique for separating the DNA fragments, that is called gel electrophoresis. Gel electrophoresis. We have the picture of gel electrophoresis here, and in this picture you can see the agarose gel electrophoresis. There is a medium taken over here, and you can see the cathode as well as the anode. The positive electrode is anode, and as you know, when you put the DNA fragments in the wells, what will happen? The DNA fragments, because of the size difference, they will move at different speeds, and you will get the DNA fragments separated here. Okay, so DNA fragments, they are getting separated based on their size. Based on their size, here in this diagram also you can see the larger fragments, they cannot move fast. Only the smaller fragments, they can move fast, and they will have displacement to a long distance. So here the fragments are separated based on their size. It is due to the charge they are moving, but their separation is according to their size. We have the question for you now. Gel electrophoresis, our answer is separation based on size. That is our correct answer. What about other options? Cutting DNA fragments and the purification. As you know, there is a step called elution. A step called elution, in which we are using ethidium bromide, and that will color the DNA, and using UV rays you can extract the DNA from the gel piece. Now, separation of DNA fragments according to electric charge, this is wrong because they are moving because of electric charge, but the separation is based on the principle or based on the idea that they have difference in their size. So, amplification of the target DNA sequence, as you know, in this chapter we deal with the PCR, polymerase chain reaction. So, this option is applicable for PCR only. Now, we move on to the next question. It is about plasmids and bacteriophages. They are used as very good vectors in recombinant DNA technology. Why? That is our question. What is the reason for using plasmids and bacteriophages as good vectors? You know, plasmids, they are obtained from bacterial cells. We have the picture now. In this picture, you can see the bacterial cell with a set of plasmids now. Okay. So here, plasmids, they will be cut with a restriction enzyme. The desired DNA is also cut with the restriction enzyme, and both of them are joined to form a recombinant DNA, and it will be introduced into the host cell. So in the question now, in this question, you can see option A, B, C, all are applicable for plasmids as well as for bacteriophages. So option A and B for plasmids, and option C is also applicable for these kind of vectors. If an alien piece of DNA linked with it, we can multiply its numbers equal to the copy number of the plasmid or bacteriophage. So all are taken directly from your textbook. So all these statements are true for vectors. Okay, that is why we are using them in recombinant DNA technology. So answer option D here. We move to our next slide. It is about genes encoding resistance to antibiotics, considered as useful dash. So here, as you know, we are using cloning vectors. We are using cloning vectors in recombinant DNA technology. And these vectors, we usually produce them artificially, and they will have these properties. And these properties are listed in our next slide. We move on to the next slide. Cloning vectors will have properties like origin of replication, selectable marker, cloning sites, and suitable vectors. The cloning vector should be suitable for plants as well as for animals. What do you mean by origin of replication? The region from where replication begins. That is called origin of replication. And what do you mean by selectable marker? Selectable markers are used for identifying and eliminating non-transformants and selectively permitting the growth of only the transformants. So here, you know, in recombinant DNA technology, sometimes the recombinant DNA, when it is introduced into a host cell, sometimes the host cell will accept that DNA. Then we say that that cell is transformed, or that cell is called transformant. But sometimes the DNA will not be taken up by the cell. Then it will remain non-transformant. So here we have to allow the growth of only the transformants, and we have to select only the transformants from non-transformants, and that is with the help of selectable markers. And usually antibiotic resistant genes, they are used as very good selectable markers. These antibiotic resistance genes will be present on the cloning vector. So on the vector we will have certain genes, antibiotic resistant genes, or genes which will give resistance against antibiotics. Genes encoding for resistance to antibiotics such as ampicillin, chloramphenicol, tetracycline, kanamycin, etc. They are considered as useful selectable markers for E. coli. So I repeat, these selectable markers are helping us for identifying transformants from non-transformants. And later on, among transformants there will be recombinants and non-recombinants. And we will be isolating only the recombinants from non-recombinants. So for that, selectable markers are very much useful. Okay, so cloning vectors, they will have cloning sites also, and it should be suitable for, the vector should be suitable for plants as well as for animals. That is meant by suitable vectors over here. So we have the next slide for you. So in this slide, we have the question: genes encoding for resistance to antibiotics is considered as selectable marker. It is used for identifying non-transformants and transformants, and also from transformants we will be selecting only the recombinants. You got it? So answer will be option B. Next question. In order to link the alien DNA, the vector needs to have a very few, preferably single, recognition site for commonly used restriction enzymes. So you know, when you are inserting the gene of interest in a vector using a restriction enzyme, the restriction enzyme that we are using should have only one recognition sequence. If there are more than one recognition sequence, what will happen? At more sites on the vector, a cut will be made and the vector will be destroyed, or the DNA is getting fragmented. If there is more than one portion where a cut is made, it will be getting fragmented. So usually in the case of a circular plasmid, only at one region a cut will be made so that we can insert the donor DNA over there. Okay. So preferably only one recognition site will be there. Okay. So we move on to the question now. We move on to the explanation slide now. So in this diagram, you can see PBr322. PBr322, it is an artificially produced plasmid. It is a very good cloning vector. And you can see the different cloning sites over here. We move on to the question again. We have the question slide. In order to link the alien DNA, vector needs to have a very few, preferably single, recognition site for commonly used restriction enzyme. Why? Because otherwise the vector will generate several fragments. So the correct explanation is given in the fifth statement. Okay, other statements are not applicable here because more than one alien DNA fragments may get incorporated. That is wrong. We are trying to incorporate only one donor DNA, only one foreign gene. Then more than one recombinant DNA will be formed. We are producing only one recombinant DNA at a time. Then plasmids may not accept alien DNA. That is also wrong. Plasmids, when a cut is made, it will accept the alien DNA if it has sticky ends which are complementary with each other. Then they cannot identify multiple sites. This is also wrong. So our answer will be option C, which says only fifth statement is true here. Now we move to our next question. It is about PBr322. So the diagram of PBr322 is very important. You should have a clear idea about the labels. So different labels are there. We have the diagram. Next slide, we have the diagram. So here in this diagram, you can see the origin of replication. In PBr322, there is origin of replication. Then ROP. What is ROP? ROP. That is the sequence which is involved in coding for proteins of required for replication of plasmid. Okay, so this sequence will code for proteins that are involved in replication of the plasmid. And you can see the cloning sites over here: PST1, PVU1, EcoRI, CLA1, Hint3, then BAMH1, SAL1. All these are PVU2. All these are cloning sites. All these are cloning sites present in PBr322. So moving on to our question, our correct answer will be option D. Because how many cloning sites are present? 8. When you count all the cloning sites shown in that diagram, total number will be 8. And what is the meaning of ROP? It is ROP is a sequence which codes for proteins involved in replication of the plasmid. Then what about selectable markers shown in the diagram? Selectable markers shown in the diagram. One is tetracycline resistance sequence and other is ampicillin. So one is ampicillin resistance sequence and other is tetracycline resistance sequence. So in this pink color, you can see the antibiotic resistance sequence or selectable markers in PBr322. Moving on to our next question. The dash resistance then gets inactivated due to insertion of the alien DNA at dash site. So this is a very important question. It is about the two antibiotic resistant sites, and it is about the site at which foreign DNA is inserted. You know, once foreign DNA is inserted into the antibiotic resistance sequence, the antibiotic resistance is lost from that cell. So the property of antibiotic resistance will be lost once a gene is inserted into the antibiotic resistant gene, and this is called insertional inactivation. To more details, for getting more details, we have the diagram. So in this diagram, first you should know about what is transformation. What is transformation? When a DNA is entering into a cell, we say that there is transformation. Okay, when recombinant DNA enters, we say that there is transformation. So during this transformation process, when we do the technique, it is not necessary that all the cells are taking up that recombinant DNA. There will be two conditions formed here. There will be a set of non-transformants, or those bacterial cells which do not take up the recombinant DNA. They are called non-transformants. So non-transformants are there is another set of transformants. They have the recombinant DNA received. Okay. Now among transformants, there will be two sets, non-recombinants and recombinants. So in the case of insertional inactivation, as I told you, there will be inactivation of the antibiotic resistant site. In the case of PBR 322, as you know, the BAM H1 region, to that region the insert is happening. So the BAM H1, it is a cloning site present in the tetracycline resistance sequence. So the tetracycline resistance will be lost in the case of recombinants. So that is what happening in the case of PBR 322 when there is insertion at the BAM H1 site. Tetracycline resistance will be lost. So all the recombinants will lose the tetracycline resistance, but they will have ampicillin resistance. So here in this diagram, it is very clear. This is about explaining about the insertional inactivation and also the selection of transformants from non-transformants, and also selection of recombinants from non-recombinants. So here you can see in the master plate there will be three types of organisms, three types of cells you can see. Some are non-transformants and some are non-recombinants and some are recombinants. So this white is non-transformant. This red one, it is non-recombinant, and this is recombinant. You got it? So here the very important point to note is that non-transformants. Non-transformants mean they don't have the recombinant DNA entered into them. So the non-transformants, they have no resistance against ampicillin or tetracycline. So they do not grow in ampicillin medium or in tetracycline medium. Okay, they cannot grow in both the media. What about non-recombinants? In the case of non-recombinants, they can grow in both the media. They can grow in ampicillin medium as well as in tetracycline medium. Okay, so what about the recombinants? Recombinants, because of the insertional inactivation, they will grow only in ampicillin medium. So here very easily you can isolate the recombinants. So all other cells will be degraded or they cannot grow in the tetracycline medium. Okay, so here we are getting only the recombinants because they only survive in the ampicillin medium. So only the recombinants finally you are getting. So this is the method of selecting recombinants from non-recombinants. You got it? So first we select transformants from non-transformants and later we select recombinants from non-recombinants. So with the help of this diagram, you will be able to understand that point. Now coming back to our question, Before that, here it is an explanation about insertional inactivation directly from your NCRT book. But here the case is different because always plotting in tetracycline medium and then in ampicillin medium, it is very difficult. So nowadays we have a new technique. A recombinant DNA is inserted within a coding sequence of an enzyme beta galactosidase. So in the coding sequence of an enzyme called beta galactosidase, we are inserting the foreign gene. So what happens? The enzyme is getting inactivated, or the enzyme coding gene will get inactivated. So no longer the enzyme will be produced. So in the presence of a chromogenic substrate, what is a chromogenic substrate? Color developing substrate. A blue color colony will be produced if the plasmid in the bacteria does not have the insert. So if the insert is absent, the colony will be colored in the presence of chromogenic substrate. Or if there is insertion, or if there is insertional inactivation, the colored colonies cannot be produced. So we can choose between cells having color and cells which do not have the color. So the presence of the insert results in insertional inactivation of the beta galactosidase, and the colonies which do not produce color, and that colony we can name them as the recombinant colonies. Okay, so this is a modern or an advanced technique of selection of recombinants. So you remember, in the new technique we are introducing the gene into a sequence which is coding for an enzyme like beta galactosidase, and we are resulting this insertional inactivation. So this is a new technique. So we don't have to plate them in tetracycline medium and later in ampicillin medium. So that difficulty we can avoid. Okay. So moving to our question now, our answer will be option A here, because they are asking about the gene, okay, and also the site. So to which gene and to which site that foreign gene is inserted in PBR322. In the case of PBR322, the gene of tetracycline, you can see the cloning site called BAMH1, and to that region we are inserting the DNA, we are inserting the foreign gene, and we are resulting the insertional inactivation. You got it? In other options, you can see the other cloning sites which are not applicable here. Okay, so in that particular example, they are talking about inserting the gene into the tetracycline resistant gene at the cloning site of BAMH1. So we have the next question. It is about the PCR. The figure shown has got 3 steps A, B, C of PCR. Select the option giving a correct identification together with that, with what it represents. So you have to identify A, B, C here. What is A, B, C here? You have the diagram. So in this diagram of PCR, you can see what is A here. A marked the, it was the denaturation. It was denaturation, or applying heat so that the strands are getting separated. Okay, denaturation step. Then labeled as B over there in that question, it was primer annealing. So second step is annealing, and the third step is extension. C, labeled as C, will be extension with the help of Taq polymerase or the thermostable DNA polymerase. Extension will be done. So the correct answer for that question will be option C. Okay. What about option B? Is it B labeled as denaturation? Can you label B as denaturation? No. Here in this B portion, it is primer annealing. So it is wrong. What about A, denaturation at a temperature of 50 degrees Celsius? No, it is not 50 degrees Celsius. Above that, more than 80 degrees Celsius will be used for denaturation. So this is also wrong. So this is the correct matching of label portion with its correct identification. Now labeled as A, annealing with the 2 sets of primers. No, labeled as B will be the annealing, primer annealing. So D is also wrong. Our answer is option C. So we move to our next question. It is about Agrobacterium tumefaciens. Agrobacterium tumefaciens, it is a very good vector for plants because Agrobacterium tumefaciens, it is having a property of inserting TA plasmid into plant cells, and this TA plasmid, it is having tDNA or tumor causing DNA. We have the diagram for you. So in the next slide, we have the diagram of Agrobacterium tumefaciens. You can see it is having the TA plasmid with tDNA over here. Okay. So normally this TA plasmid will be inserted into the plant cell and the plant cell is getting transformed. But here in our biotechnology, what we do, we are inserting the gene of interest. So the DNA with the gene of interest, it will be incorporated to the tDNA portion, so that we are introducing the desired gene into the plant cell, and you can see the plant with a new trait, a plant with a new character formed. So Agrobacterium tumefaciens, it can be used as a very good vector for plants. So coming to the question now, in this question our answer will be option B. So tDNA belonging to TA plasmid, that is our answer for this question. What about the other options? They are not relevant here. RA plasmid is resistance inducing plasmid, then F plasmid, fertility plasmid, so col plasmid, colicin producing plasmid, cDNA, by cDNA we mean complementary DNA, ssDNA means single stranded DNA. So all these options are only for confusing you or for distraction. Our answer will be option B only. Now coming to our next question. Select the method of vector mediated gene transfer. So you know gene can be transferred using vectors, that is vector mediated gene transfer, and there are other techniques in which without vector also we are introducing the gene of interest into a cell. So that is called vectorless gene transfer. What are the different types of vectorless gene transfer methods? One is microinjection into the target nucleus, that is vectorless gene transfer. Then chemical mediated gene transfer is also another method of vectorless gene transfer. Then biolistics we mean the gene gun method. And electrophoresis, by using electric pulses, we are creating some pores in the cell membrane so that the DNA can be inserted. So all these options B, C, D, they belong to vectorless gene transfer. Only the vector mediated gene transfer will be option A here. So here we have the explanation slide with the electrophoresis process. Electrophoresis, can you see that? Electrophoresis here. So electric pulses, they can create some pores and the DNA will be introduced into the cell. Okay. And now here in this portion you can see the gene gun method or biolistic method, where DNA coated onto the some pellets, gold or tungsten can be used as pellets here, and they are literally shoot into the target cell. So that is a gene gun method or biolistics method. Okay, now coming to our question now, our answer will be option A. Cosmid mediated gene transfer, that is a different one. You know what is cosmid? Cosmid, explanation of cosmid was in that previous slide. We will go to the previous slide. What do you mean by cosmid? Cosmid is having a feature like this. It is having a core site. That is why it is called cosmid. So it is a special kind of plasmid with a core site. That only you have to note. Okay. So other features explained here in detail. So you just remember that it is having core site. Okay. Now our answer is option A. We move on to our next question. Which is our next question? It is match the following question. You know, in the process of recombinant DNA technology, we have a step called isolation of the DNA. Isolation of the DNA, for that we are using different enzymes. If you are using plant cells, we need cellulase to degrade the cell wall. If you are using bacterial cell, you need the lysozyme enzyme. If it is a fungal cell, chitinase is required. If it is RNA, there you have to remove that. You have to take only the DNA. So for that, RNAs or ribonuclease is required. Then proteases, sometimes proteins you have to remove. Then you need the protease. Sometimes with the DNA there will be protein content, so proteases are used. So all these enzymes are used in isolation of the genetic material. And ethanol, finally, by adding chilled ethanol, we do the spooling step in isolation of the genetic material or isolation of the DNA. Spooling step require chilled ethanol. So for DNA extraction, finally, purification, finally, we need chilled ethanol also. So, which is our correct answer? We have the next slide showing spooling technique. We move on to our answer slide now. Our answer will be option A here after matching. Now we have the next question. It is about thermostable DNA polymerase. You know what is a thermostable DNA polymerase? In the denaturation step of PCR, we require high temperature, and in that high temperature, many of the enzymes will be denatured. So for PCR, we have to use thermostable enzyme. And which is the thermostable enzyme used here? Taq polymerase, which is isolated from Thermus aquaticus. So Taq polymerase is isolated from which organism? Thermus aquaticus. So that is used in PCR or polymerase chain reaction because it can survive at a very high temperature. It is usually present in hot springs. This Thermus aquaticus is a bacterium which is present in hot springs. That is why we can use that particular enzyme in PCR. So correct answer is option B. To explain other options, we have the slide for you. So in this slide, you have the explanation of Thermus aquaticus. It is written clearly that it will survive 50 to 80 degrees Celsius. Okay, it will best grow at 65 to 70, but it can survive a temperature of 50 to 80 degree. And also regarding Salmonella typhimurium, what you can say about Salmonella typhimurium? It is used to investigate its ability to deliver DNA into the antigen presenting cell in order to produce cancer vaccines. So Salmonella typhimurium, in the beginning of your chapter, you say that Salmonella typhimurium, it was used for taking the plasmid for producing the first recombinant DNA by Stanley Cohen and Herbert Boyer. So there we mention about Salmonella typhimurium. What about Pseudomonas putida? Pseudomonas putida, these strains are capable of degrading petroleum hydrocarbons, and they can be used, they can digest the hydrocarbons. So we can use them to clean the oil spills. To clean oil spills, we can use Pseudomonas putida. And what about Pseudomonas fluorescens? We are using in our agriculture purpose this Pseudomonas fluorescens because it is having some ability to of bioremediation. What do you mean by bioremediation? This can be used for checking the growth of fungus, plant fungus. So that is the use of Pseudomonas fluorescens. It has shown that microbes potential benefit in bioremediation against several strains of plant pathogens. High concentration of all 5 strains of Pseudomonas fluorescens tested inhibit spore formation, spore production by pathogenic plant fungus. So it is able to check the fungal growth in plants, Pseudomonas fluorescens. So with this, we have the next slide. With this, we conclude today's session on biotechnology. So we have one more slide. With this, we conclude today's session on biotechnology. So tomorrow we will be back with a topic from your first year syllabus. We will have breathing and gas exchange, questions from breathing and gas exchange. So meet you tomorrow at the same time. Until then, it's goodbye from Stephen Classes.